My dear students of 12th class, today in your botany we are going to take up packaging of DNA helix. As you know, the DNA being a molecule with a very huge molecular mass, and how it is packed within the nucleus remains a big deal to be explained. And this packaging of the DNA helix requires a proper mechanism by which we will expect the DNA to get inserted inside the nucleus. And now, if we take the distance between the two consecutive base pairs as 0.34 nanometer, 0.34 nanometer is multiplied by 10 raised power minus 9 meter. That is 0.34 nanometer equals to 0.34 into 10 raised power minus 9 meter. And you can understand how smaller the unit of nanometer is. If the length of DNA double helix in a typical mammalian cell is calculated simply by multiplying the total number of base pairs with distance between two consecutive base pairs, that is 6.6 into 10 raised power 9 base pair multiplied by 0.34 multiplied by 10 raised power minus 9 meter per base pair, it comes approximately equal to 2.2 meters. And this you can understand, the length of the DNA is approximately 2.2 meters that is to be packaged within the nucleus. You can imagine the structure of the cell being such a minute creature, and the nucleus is so tiny, how it is capable of holding such a huge molecule with a length of almost 2.2 meters. A length that is far greater than the dimension of a typical nucleus, that is approximately 10 raised power minus 6 meters. How is such a long polymer packed in a cell? In prokaryotes such as E. coli, though they do not have a defined nucleus, the DNA is not scattered throughout the cell. DNA being negatively charged is held with some proteins that have positive charges in a region termed as nucleoid. The DNA in nucleoid is organized in large loops held by proteins. That was about the prokaryotes. Now, in eukaryotes, this organization is much more complex. There is a set of positively charged basic proteins which are called as the histones. Again, in eukaryotes, the organization is much more complex, where there is a set of positively charged basic proteins called histones. A protein acquires charge depending upon the abundance of amino acid residues with charged side chains. Histones are rich in the basic amino acid residues lysine and arginines. Both the amino acid residues carry positive charges in their side chains. So, the lysines and the arginines, both the amino acid residues, carry positive charges in their side chains. And histones are organized to form a unit of 8 molecules called a histone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. This you can visualize here on your monitor. This is the how a histone octamer is wrapped over by the DNA, and together this is called as nucleosome. And now, nucleosome, a typical nucleosome contains about 200 base pairs of DNA helix. Nucleosomes constitute the repeating unit of a structure in nucleus called chromatin, which is thread-like strand-colored bodies seen in the nucleus. The nucleosome in chromatin are seen as beads on string structure when viewed under electron microscope. The nucleosome in chromatin are seen as the beads on a string when they are viewed under an electron microscope. Now, theoretically, how many such beads can be imagined to be present in a mammalian cell? That depends upon the number of the base pairs that a particular nucleus contains, rather the particular DNA molecule of a particular organism has. The beads on the string structure in chromatin is packaged to form chromatin fibers that are further coiled and condensed at metaphase stage of cell division to form chromosomes. The packaging of the chromatin at a higher level requires additional set of the proteins that are collectively referred to as non-histone chromosomal proteins, NHC. And in a typical nucleus, some region of the chromatin are loosely packed and stain as light and are referred to as euchromatin. And the chromatin that is more densely packed and stain as dark is called as heterochromatin. A euchromatin is said to be transcriptionally active chromatin or the true chromatin, whereas the heterochromatin is the inactive part of this chromatin material. Thanks. Have a nice day.